Euplotes. The species illustrated here seems to be Euplotes eurystemus. It has a rather distinct anterior rostrum and other typical features of the species. Many of its identifying features were discussed in detail in Part 1 on euplotes. The German term Kern or Kern is used here to refer to the nucleus. In this photomicrograph, the organism can be seen to have a nucleus which takes the form of a thick strip or semi-twisted band, hence the term bandiform nucleus. The page on the viewer's left is taken from Call's text of 1933. The nucleus of euplotes is commonly in the form of a horseshoe or the letter C or a kind of loosely drawn Arabic numeral 3. In euplotes eurystemus, the nucleus tends to be large and bandiform and to take on the form of the Arabic numeral 3. This form of the nucleus of euplotes is not limited to the species eurystemus, and as can be seen here in the diagram from Call, it is also seen in the species Affinis. Although it is not always visible, euplotes has a micronucleus, which is usually closely applied to the nucleus. It takes the form of a large, dark staining, dot-like structure. Patterson, in his textbook, makes mention of the undulating membranes, which are to the right of the mouth in euplotes. The next few screens will illustrate the locale and movements of these membranes. Because the organism is transparent, these membranes and their activities are visible from the dorsal aspect as well as the ventral aspect. In the inset view here, on the observer's upper right, it can be seen that the membrane is quite delicate and that it overlaps the distal AZM, with which its movements can be confused. The fused distal cilia of the AZM also undulate. In the distal region of the ventral AZM, there are two pillar-like structures with a narrow space between them, which progressively narrows distally. This is the entrance to the cytostome. Here in this lateral view, euplotes is perched, hanging by its anterior cirri on a local piece of vegetable matter. The arrow draws your attention to the flow of particles in a stream which probably results from the activity of the AZM and associated membranes. The cytostome is selective and does not necessarily accept all particles which are presented. The second arrow points to the location of the contractile vacuole, which can be seen to both fill and empty during this video. This video provides a ventral view of the contractile vacuole of euplotes. Note that in the ventral view, the contractile vacuole is on the organism's right side and it is beyond the midline of the body.